Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Dawn Kernagis, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the Double Secret Selection Committee that selects all the guests to appear on STEM Talk.
Hi, Dawn. Great to be here with you. Today's interview with Dr. Suzana Herculano-Houzel was not only fascinating, but as you will hear, it was fun.
It was a lot of fun. And Suzana is a Brazilian neuroscientist who devised a way to count the number of neurons in human and animal brains. She calls it brain soup.
Bon appetit.
Ooh.
She writes about this in her book, The Human Advantage, a new understanding of how our brains became remarkable. Her method of counting the neurons of human and other animals' brains allowed her to study the relationship between the cerebral cortex and the thickness and number of cortical folds in the brain.
Before we get to today's interview with Suzana, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we are especially appreciative of all the wonderful 5-star reviews that are piling up on iTunes. As we announced in several earlier episodes, the Double Secret Selection Committee has been continually and carefully reviewing the iTunes reviews, with an eye towards selecting the wittiest and lavishly praise-filled reviews to read on STEM Talk. As always, if you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Really cool shirts. Today, our winning review was posted by someone who goes by the nickname of T-ville Flash, and who was commenting on episode 31, which was an interview with theoretical cosmologist Michael Turner, he who coined the phrase dark energy. The review is titled, "Nothing Dark About This Podcast." T-ville Flash wrote, "I hope all science teachers take the time to listen to this podcast and share it with their students. Michael shares a story about how great high school teachers led him into science. But more importantly, he eloquently explains why young people ought to pursue science careers. I love the part where he said, 'I get to think about the mysteries of the universe.' That's a pretty good job to have." And after listening to this podcast, it does actually sound like a great job to have.
Well, thank you, T-ville Flash, and thank you to all the other STEM Talk listeners who have helped STEM Talk get off to such a great start.
Okay, now on to today's interview with Suzana Herculano-Houzel.
Dr. Herculano-Houzel is an associate professor of psychology and biological sciences in Vanderbilt University's Psychological Sciences Department and the Vanderbilt Brain Institute. She grew up in Brazil and received her undergraduate degree in biology at Federal University of Rio de Janeiro. She went to Case Western Reserve University in Cleveland, Ohio to get her master's in neuroscience and completed her PhD in visual neurophysiology at the Max Planck Institute for Brain Research in Frankfurt, Germany. After completing her doctorate, Suzana returned to Rio and went to work for the Museum of Life, where she designed children's activities. In 2002, she returned to her alma mater and began researching how human brains compared to other animals.
Last year, MIT Press published her book, The Human Advantage, a new understanding of how our brain became remarkable. The book tells the backstory of her research on human and elephant brains and how she came up with brain soup. In 2004, she devised a way of reducing brains to liquid as a means to count the number of neurons in them. It is technically known as the isotropic fractionator. Prior to her research, scientists assumed that brains of all mammals were built in the same way and that the overall brain mass as compared to body mass was the critical determinant of cognitive ability. It was to resolve these conundrums about brain mass, body mass, and intelligence that Herculano-Houzel turned to chainsaws, butcher's knives, and kitchen blenders to concoct her brain soup.
In 2004, she won the Jose Reis Prize of Science, and in 2010, she received the James S. McDonald Foundation Scholar Award in Understanding Human Cognition. She is also the author of a bi-weekly newspaper column on the neuroscience of everyday life in the major newspaper in Brazil. Going into its 11th year, the column has appeared more than 270 times since 2006. In addition to The Human Advantage, Herculano-Houzel is also the author of 6 books in Portuguese that focus on the neuroscience of everyday life. She also has a popular blog called The Neuroscientist on Call, which she describes as not-so-random thoughts about brains, the universe, and everything. Suzana lives in Nashville, Tennessee with her husband, son, and 2 dogs.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
STEM Talk.
Hi, welcome to STEM Talk. This is Dawn Kernagis, and I want to welcome our guest today, Suzana Herculano-Houzel. Suzana, welcome to the podcast.
My pleasure. Thank you so much for having me.
Yeah, and also joining us today is IHMC's director, Ken Ford.
Hi, Dawn. Great to be here. And hello, Suzana.
So, Suzana, let's start at the beginning and discuss when and how you first became interested in science.
I think I always had a keen interest for how things worked. And I remember that living things in particular were always very interesting. You know, how come they move and shine and change shape, and how does that all happen? I don't think I was aware that I was interested in science as such from the beginning. I do remember that my parents played a big role in that. They were always encouraging me to pursue questions. And I think a big thing that pays off now in what I do is they always encourage us to question authority, you know, not take people's words for a fact, just be free to inquire about how things work and how do we really know that this is what it is, and how do we know that this is how we think it works. And I think in the end I realize now that really is a major driving force, let's say, of scientific inquiry. It's really just being inquisitive about how things work and how the world is, and going for it and just taking the leap and investigating it. It's a lot of fun.
Yeah, absolutely. Getting paid to be curious is a fantastic position for sure. So from what I understand, so you grew up in Rio, and I'm guessing that you loved it because after you got your doctorate, you returned back home to Rio and went to work for a science museum. So what kind of work did you do at the museum?
Well, I had spent 7 years abroad, and I remember I left Brazil very early. I was 19 when I left home, and I remember the feeling of not wanting to feel like I was in exile anymore. Family is a big thing for Brazilians. It was nice to have an opportunity to be back home with my family. I also wanted to start my own family. My kids were born very soon there, but yeah. So I had realized at that point that I enjoyed reading about different topics very much. I could also write easily about them. And at the same time, I also realized that I didn't want the traditional postdoctoral position where you become super specialized in one particular topic. I enjoyed so much reading about different things that I figured, well, I'll just try my hand at a job with scientific communication. And I knocked on the door of this new science museum in Brazil. It was called the Museum of Life. And it's really a matter of being shameless about it, because come on, the worst that can happen is they'll say no, right? But I just knocked up on their door and say, hey, I just got a PhD in neuroscience. I wonder if you would be interested in having a neuroscientist in your staff. I could come up with activities for your public. So they loved it. I got really lucky. So I started creating hands-on activities for the general public. We had like a hands-on 1 hour long science show. It was a lot of fun. And I also started writing my own website. I created my own website. It was called in Portuguese, the version of Our Daily Brain. And that was the idea, writing about neuroscience aspects of everyday life, how neuroscience research can be informative about our everyday life, the world, how we think, how we make choices. And I was very concerned in the beginning that I would run out of subjects. But I very rapidly realized that was not a problem. I kept a pile of papers. I always wrote based on recent publications, recent scientific publications, and I kept a stack of papers that were of interest until I realized there was no point. I had no risk of running out of subjects.
Yeah, I can imagine, especially in neuroscience.
Yes, absolutely. You must really enjoy writing. You ended up writing a regular science column for the newspaper as well, and for Scientific American Brazil.
Yeah, I still do write for Folha de São Paulo. That's a major newspaper in Brazil. I started writing. Well, yeah, that was 2002. So I started writing 15 years ago, and it's something I learned on the job. I never had any formal training to do it, but I enjoyed it very much, and I think I quickly learned that if you put yourself in your audience's place, if you think of what do they not know that I need them to know to be able to grasp this particular concept that I want them to be able to grasp, it helps a lot. It makes things much easier, and also it avoids what I always disliked about so many popular books on science or neuroscience for the lay public, which usually begin with, well, I'm going to tell you a bunch of interesting things, but hold on. First, I got to explain to you what a synapse is. And first, I got to tell you what a neuron is. And I was like, no, just tell them the story already and give them the necessary bits as you go. And the real fun is that once you do that, people realize that they can understand it, and that is so rewarding to them. And in return, I started getting these emails from people saying, I feel so smart. I could finally understand something about the brain. Please keep writing. So that was probably the biggest reward I could ever get. So of course I kept writing, and I still do that every 2 weeks for the newspaper. I wrote for Scientific American Mind for, I think it was 4 years or so. But I like writing for the newspaper. It's short pieces, and I have leeway to write about whatever I want. So a few years ago, I started referring to myself as the neuroscientist on call. You know, it kind of becomes a job hazard that whatever I do, I'm walking around, I'm with my family, I'm in the movies, the back of my mind, I realize is thinking of how neuroscience fits here or there, or how there's a really cool story I could tell about this or that.
That's fantastic. And it takes a special gift to not just do the research, but to be able to get that out in an interesting way to the lay public. So it's fantastic that you're doing that. So as you transitioned into academics, were you always interested in neuroscience specifically?
No, I left Brazil to start graduate school at Case Western Reserve University here in the U.S. And the idea was that I would get a degree in genetics. I did not know the first thing about brains. That was a huge gap in the curriculum at the university I attended in Brazil. I never had any courses about brains, neuroscience, neuroanatomy, neurobiology, anything. So I got lucky that I made friends with this guy, Ken Tovar, who had been in neuroscience already. He was also in the graduate program, but he had worked as a research assistant in a neuroscience lab beforehand. And I remember doing the first of the required rotations that we had to do in the program there. And he asked me, could I try to talk you into changing to neuroscience? And I'm like, sure. Talk ahead, talk away, just tell me. And we would start chatting about, the most random things. And he was capable of pulling in bits of knowledge about neuroscience and these really cool questions. I remember walking on the street with him, and he would just stop in front of a tree with the tree behind his back, and he would just reach out to his back, and he was like, how do I know it's here? I can see it, and yet I know it's right here. See? And he would just tap at the tree behind him. It's right here. Mm-hmm. Somehow my brain is able to create a representation of things that are not around. And I remember going, whoa, this is so cool. So I took the last mandatory rotation in a neuroscience lab, and I just started learning about that new world. And the more I learned about it, the more I wanted to hear about it. The really interesting thing, I think, that caught me was that, and this is no demerit to genetics. I think it's wonderful that a lot of people think it's really the coolest thing to work in molecular genetics. But I found myself working with little tubes that had transparent liquid that I had to believe and run the tests that told me that they actually had what I thought they had. And I was becoming more and more specialized about that little part of that little gene, in that little chromosome. And all of a sudden, there was a whole new world out there that didn't end at the lab's door. I could just walk out of the street and keep wondering about all sorts of things. And that was the main hook for me. It's unending topics, unending subjects and questions.
So I'm going to transition a little bit into the specific work that you've been doing. And so I know that you said you like to kind of develop a context and a story and tell a story about certain specifics when it comes to the brain. So not just talking about the synapse or talking about different types of cells, but just so we can get an overview of the different types of cells that we're talking about in the brain. Can you provide kind of a general description of the cells that make up the brain?
Sure. The brain is an organ made of various tissues like any other organ in the body. And this particular tissue that makes up the brain is made of different cells. So there you have neurons, which are considered the basic information processing units of brains and nervous systems. Around the neurons, you have glial cells of different types. They used to be considered just scaffolding or just stuffing really around the neurons until some 10 years ago or so, people started realizing that they do so much more than that, including teaching neurons to form synapses, so the points where the places of actually not contact, but where they, without touching, they exchange information and you get signals passing from one neuron to the next. So glial cells are the cells that instruct neurons to form those synaptic not contact, but the synapses where they exchange information. And they also, the glial cells also provide neurons with metabolic support. So they not only instruct neurons to form synapses, but they also provide neurons with the energy that supports synaptic activity. So you can't really have a functional adult brain without glial cells. Now, the interesting thing is that very early in life, probably even when we are born or right before it, we do have a brain that's essentially neurons only, without glial cells. Now, besides all those cells, there is a third cell type, and that's your vasculature. That's the endothelial cells that compose your capillaries. And we usually forget about them, but again, occupational bias once more. I'm starting to become more and more interested in the capillaries that form the brain, exactly because they are the source of all energy that keeps neurons working and keeps brains alive. So that's a really interesting question to me. How much energy do different brains cost, and how does that relate to how many neurons you have and how many synapses you have? But anyway, yeah, that's brain tissue. It's neurons, glial cells, and the vasculature that keeps everything alive.
Oh, fantastic.
It's often been noted that when comparing across species, the observed differences in intelligence cannot be accounted for only by size. The best science sometimes depends on asking what at first blush might sound like the most basic of questions. And your basic question seems to be, how many neurons are there within a brain?
Yeah, and you know what? I was really surprised when I first got interested in all these questions about 15 years ago. And I was really surprised to realize that we actually didn't know the first thing about what different brains are made of. It was interesting. It felt like everybody thought that everybody else already knew. You could find things in textbooks like the human brain is made of 100 billion neurons and 10 times as many glial cells. But when I started digging through the literature for the original data, whoever said that we have 100 billion neurons and 10 times more glial cells, that questioned the authority thing my parents had going, where does this information come from? I realized that nobody actually knew, not about human brains and much less about other brains. So part of it was because there's apparently a game of telephone running in the scientific community where a small piece of information for a small structure got actually confused for numbers for the whole brain, or an order of magnitude estimate, which to me is what the 100 billions was. It was an order of magnitude estimate without any actual full factual numbers, but that order of magnitude estimate got mistaken for the real thing. And they're round numbers. They sound really pretty and really good. 100 billion neurons, 10 times as many glial cells. Sure, why not? So we had gotten to the point where you could get away with a statement to that effect in the opening lines of a review paper or a chapter in a textbook, and nobody would ask you for the evidence. But I started reading through it, and I think it's kind of curious. What got me interested in all of this, what got me started in everything that we're doing these days, was that other myth, this one much more popular, I think, that we only use 10% of our brain. You've probably heard that one. Long story short, I still have no idea where that came from, but I started reading around because I ran a survey when I started working at that museum in Rio. I wanted to have a feel for what people thought about the brain, and I realized that 60% of college-educated people in Rio thought that they went about life only using 10% of their brains. It's like, what a huge waste. You really think 90% of your brain is just dead weight that's really not doing nothing? So it turns out we use, of course, the whole brain at all times. It just functions in different ways. But, and I still don't know where that came from, but it turned out that one possibility, and I did find a couple of mentions explicitly to this, was that, well, if we have 100 billion neurons and 10 times as many glial cells, then neurons are roughly 10% of the cells in the brain. So I started wondering, is this how at least some people got the idea that they only use 10% of their brain cells? So that's what got me digging through the literature, looking for the original references for how many cells human brains are made of, and also other species. And that's where I realized that we really didn't know the basics. Neuroscience was doing all the super fancy, really cool work on studying molecules and pathways and genes and expression and function, but we actually didn't know the first thing about what different brains were made of. And especially, I realized that there were a number of intuitions in the field about how different brains compare, intuitions that a larger brain should always have more neurons than a smaller brain, that two brains of the same size should always have similar numbers of neurons. And it was because of intuitions like those that people started developing this concept that the human brain had to be special, essentially because we don't have the largest brain around. And yet we like to think highly of ourselves. We like to think that we're the only ones studying other forms of life and not the other way around. I think it is a fact that we are the species that has become the most capable of modifying their own environment, for better and for worse. Because of all those intuitions about what different brains were made of, and that larger brains always had more neurons, there was this notion in the literature that human brains were special in the sense of being outliers, of being unlike everything else. The rules, the basic rules of biology and evolution do not apply. And that's where this question of the authority thing just came to mind, really, because it seemed, with my background in biology and evolution, it just seems so unlikely that, the rules of the world apply to every living being but us. Not humans. We're special. Sorry, we're rules don't apply here. It seemed really unlikely, and that's what got me started. That's what got me interested in figuring out what I thought were the basics. What are different brains made of? What are the rules? How do different brains of the same size or different sizes, how do they compare in numbers of neurons, especially if you consider that neurons are these basic information processing units of different brains? And it turned out that, sure enough, a number of those intuitions were wrong.
From a computational perspective, your view makes eminent sense. One certainly would not weigh a computer to ascertain its computational power. In general, the computational power of a chip is directly correlated with the number of transistors. So it's quite analogous to the perspective that you've so nicely laid out.
Yeah, I know, and I love that analogy. The most powerful computers we have these days were so much smaller than the first huge machines that we had, right? So it really can't be size. I mean, how could we have been misled for so long to think that size must really be the most important feature? Something we've learned recently is that birds with those tiny little bird brains, they're actually deceiving in their numbers of neurons. You can, if you take a parrot or a crow, they have brains about the size of a walnut, and they still pack even more neurons than you find in the brain of a primate that's much larger than that. So it really doesn't seem to be about the size of the brain, or actually even about the size of the brain relative to the size of the body, which is something that used to be a common argument about what makes the human brain special. Because compared to other species, we supposedly have a brain that's much larger than it needed to be, let's say, for the size of the body that we had. But the more we look at different species, the more I think that the size of the body is actually mostly irrelevant. Let me give you another example. We became interested in studying crocodiles because they keep growing throughout life. And the major, again, intuition was that whenever you have a larger body, you need more neurons to operate that larger body, which again makes perfect sense, right? I figured if we look at crocodiles and we know that they have ongoing neurogenesis throughout life, that means their brain is capable of generating new neurons throughout life. I figured, well, then we should be able to figure out if larger crocodiles really have more neurons in their brain. And it turns out that they do, but only exactly in those parts of the brain that have nothing to do with processing information directly from the body. Those structures in the crocodile brain that process body-related information right away, those keep the same number of neurons throughout life. And actually, it's a ridiculously small number of neurons. The crocodile brain as a whole, even when you take a 90 kilogram, a 200-pound crocodile, a huge, massive animal, it still only has as many neurons in the brain as a rat with a tiny little body. See, so I think that illustrates the point that I want more and more to make, that it's really not about the body. It takes surprisingly few neurons to operate a body.
Having had several interesting interactions with alligators and crocodiles, I do not find this information hard to believe.
I was just thinking that we need to move away from calling people bird brain to calling them crocodile brain.
It's a compliment. Calling people bird brains is actually a compliment.
It's
yes.
a great thing, yeah.
Brains represent about 2% maybe of the average human's mass and account for roughly 25% of our energy requirement. And I wondered if you could briefly talk about energy use in the brain.
Sure. So that was the other way in which the human brain seemed to be special, literally extraordinary, right? That it is so small relative to the body as a whole, and yet it chugs this massive amount of energy. Well, but it turns out that once we had the ability to estimate numbers of neurons in different brains of different species, we could pull out data from the literature on how much energy different brains cost. And again, it's surprising how little data there is out there. But there was enough there on 6 different species that we could see how that related to the number of neurons in the brain, whereas everybody until then had only been able to work with brain size, right? And there you get this, what seems really amazing, because per gram of tissue, the human brain actually costs less energy than a mouse brain, which sounds really weird, right? Because we like to think of our brain as being this really active, massively energy chugging, super capable machine. So you would expect that it cost more energy per gram of tissue than a mouse brain. But it turns out that once you have numbers of neurons, you rapidly realize it's really simple math, because it turns out to be a linear relationship, that the amount of energy that a brain costs seems to be a simple linear function of how many neurons it has. So compared to these other species that we could analyze, the few species that there were data available for, what we found was that the human brain costs exactly the amount of energy that you would expect for its number of neurons. It's about 6 kilocalories per billion neurons per day. So it's simple math that you can do. And so it turns out that the human brain costs exactly what you would expect it to cost for its number of neurons, regardless of how large the brain is, regardless of how large the body is. And the reason why our brain costs proportionately so much compared to the body is that we're a primate. And that actually is the case for any other primate, supposedly, that a primate brain costs proportionately a lot of energy. And that's simply because compared to other mammals, to other non-primate mammals, primates have many more neurons in the brain for a given body size. So that means take 2 animals of about the same size, say 1 kilogram, 10 kilograms. One is a primate, the other is not. The body has the same size, but the primate brain actually has many more neurons than the non-primate brain, even if they have roughly the same size. So because it has more neurons, it of course costs more energy. So it turns out the human brain is really energetically costly, but simply because it has a very large number of neurons.
Fascinating. So I'm kind of curious about this whole bit about counting neurons. So I can tell you, as a previous, student in the lab, both at grad school and undergrad, and counting plenty of cells in cell culture, I'm curious about this approach, and I'm sure our listeners are curious about this approach for counting the number of neurons in an entire brain. So you've developed this unique way to count neuronal cells that you refer to as brain soup, which I love. It gives you this great image. Actually, we saw images that were sent from one of our staff members who was in your lab and could send us some pictures of the brain soup that you have stored in the refrigerator. Hopefully they're appropriately labeled. Can you please expand on this method for that you've developed and compare it to previously used methods for counting neurons?
Sure. So the traditional way of counting neurons is you take the brain, you slice it really thinly, so you make brain carpaccio to stick to the culinary analogies. And then you take some of those really thin brain sections and you stain them so you can see the neurons, and you take them to the microscope. And essentially, you're trying to determine how many neurons you have per small fraction of that tissue. That works perfectly fine as long as you stick to a given structure that has a similar, let's say, homogeneous distribution of neurons, a homogeneous density of neurons. The problem with using that approach, which by the way is called stereology, the problem with using that approach to the whole brain is that whole brains are extraordinarily heterogeneous. You have a certain density of neurons, a very high density of neurons right here, but you move 2 millimeters to the side and you have a very neuron poor region, and move 2 more millimeters away and you have a whole new structure that has a whole different type of density of neurons, a whole different organization. So if you were to use that approach for the whole brain, you would have to start by, let's say, parcellating that whole brain down into probably hundreds of structures, each with their own different density. Now, that is, of course, impractical. And it was all the more impractical to me because 15 years ago, when I became interested in these questions, I was a professor at the university in Rio, but I had no lab. So forget about a fancy microscope and the fancy software that it takes to do stereology. But I realized, reading literature from the 1970s, that there had been this attempt to estimate total numbers of cells in different brains by dissolving the whole brain, collecting DNA, extracting DNA from the whole brain, and then dividing that total amount of DNA by the estimated amount of DNA per individual cell. Which, of course, there will be a lot of errors in the calculation because the final numbers will depend on how accurate both your extraction and the DNA content per cell were. But I remember reading those papers and going, no, don't count DNA, count nuclei. As supposedly, that's the one basic assumption of the brain soup method is that every cell in the brain has one and only one cell nucleus. I mean, that's basic high school biology, right? And so that's when I realized that if I could literally, physically dissolve the tissue in detergent, if I could harvest all the nuclei, and it turned out that I could do that very easily, provided that the tissue had been well fixed before, well preserved, that made the nuclear membrane so incredibly resistant to mechanic friction and forces, that I actually could collect all the nuclei in a suspension, in a soup, really. It actually looks very much like unfiltered apple juice. That's what my students like to tell me, that I had killed apple juice for them. But that's really the essence. You can do it with you can do it with a whole brain, but you can also use this method. You can also turn into soup any part of the brain that you can dissect. So any structure that you can dissect, you can count separately. And the beauty is that it's very fast. You get an estimate in 10 minutes. And this estimate is completely independent of the size of the structure, because you determine what the final volume of this suspension of this brain soup will be. And because it's a soup, now you don't have that problem of heterogeneity anymore. You've literally dissolved the heterogeneities in the distribution of cells in the brain. So any 4 or 5 samples that you take out of that suspension, you can take to the microscope and count them, and they will be representative of how many nuclei you have per volume of the structure.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
Last November, the New York Review of Books gave your book, The Human Advantage, an excellent review. At one point in the review, the reviewer noted that, I love this quote, you turned to chainsaws, butcher's knives, and kitchen blenders to make brain soup. Did you have any idea that this question you were asking was going to require you to take up chainsaws, butcher knives, and kitchen blenders?
You know, when I started working on this, and it was in a collaborator's lab in Brazil, Roberto Lenz, I was still devising how to turn brains into soup. And I remember that for whatever weird reason, he had a kitchen blender on the top shelf in the lab. And I remember walking into the lab, looking at that kitchen blender and thinking, am I really gonna take a human brain, something that used to be the essence, the very core of a person, and just pulverize it in a blender? Which sounded horrible, right? It sounds really dramatic and like final. But the thing is, if you think about it the right way, it's not that far. And also you can do the much gentler process in the end. We use nothing like kitchen blenders. But what we still do to study brains in general is to take those brains and slice them really thinly, like the carpaccio was talking about. And then you take those sections and you look down at the units that make up the tissue. So in a way, dissolving brains into soup is really just going one more step further. It's really, rather than stopping at the level of here we have individual cells that we can see, it's the next level of dissolving the cells and looking at the nuclei, and it serves a purpose beautifully of allowing you a much easier handle on the numbers that compose the tissue. So no kitchen blenders, But yeah, I had no idea of what lay ahead, that there were chainsaws and butcher knives in my future. I like to toy with the idea of having an extra section in my CV called other skills. and that's where I could put, can cut fillets out of crocodiles and antelopes, can do precision work with a chainsaw because that's what you need to open the skull and be able to collect the brain, right?
You could have a sort of culinary section in your CV.
I don't know how well that would play, Ken. Yeah.
Suzana, could you talk a little bit about your struggle in the early days of your research to locate available brains?
The thing is, the first part of the work was straightforward because first it was developing the method itself, and that I could, of course, do with, and I should preferably do with lab animals, with mice that we could easily raise in the lab, and rats and other small rodents, guinea pigs, hamsters. That was all within reach. Those are still regular lab animals, let's say. But the thing is, the more I realized that we not only could get numbers, but they were meaningful and they were starting to tell a different story. I mean, we could actually, for the first time, get actual numbers for different species, including human brains, and, just check, really verify the basics of neuroscience. The more I realized that we could only do this properly if we went for full diversity. If the problem is that I later realized there were a number of studies out there that were interested in looking at diversity, were interested in comparing the human brain to other species, but for obvious practical reasons, they were usually limited to looking at mouse and rat, and then a monkey brain. Maybe if they were lucky, they had access to preserved chimpanzee tissue, and then human brains. So the comparisons that we had were really between humans and macaques and mice. So that's very much like comparing apples to oranges. I mean, the proper comparison would be, well, how does the human brain compare to another similarly large brain that will, of course, not be a primate brain? So I realized that we needed to address as many different brains as possible, starting with mammalian brains. So in the beginning, that was hard because, there's very few people in the world who have the interest, much less the access to brains of different species. But things started moving first when I met John Kaz, who's a professor here at Vanderbilt University. We started collaborating 11 years ago because he learned about our work on rodents, and he became instantly interested in doing the same thing with primates and finding out how primate brains compare to rodents. And that's when we learned that primate brains are made differently from rodent brains. Per unit size, per gram of tissue, you find many more neurons in a primate brain than in a rodent brain. Of the same size. So that's when we started realizing that the basic intuition was wrong. You do not necessarily have similar numbers of neurons in brains of the same size. That was the second research that we published, first on rodent brains and then second with John on primate brains. And it was interesting because in the beginning there was a lot of pushback, right? Who's this person coming out of nowhere, turning brains into soup, for heaven's sakes, and arguing that primate brains are different from what we expected, that primate brains are made differently. It kind of got worse in a way when we published the data on human brains, because there the message was, we're just a generic primate in at least in what concerns numbers of cells. We have exactly as many neurons as you would expect for a primate with a brain the size of ours. Some people could just not have that. But anyway, the good thing was that that made it easier to convince the few people out there who did have the means and the interest to look at different brains, different species. And one, I was really lucky to get in touch with Paul Menger. He's a comparative neuroanatomist in South Africa. His main focus is neurochemistry. And so we have really complementary interests in looking at brains of different species. And we started collaborating about 7 or 8 years ago on first with elephant brains, and actually it quickly turned into much, much more than that because Paul has this, besides other things, he has a love for putting his Land Rover out in the savannas and just driving to places and collecting the animals and the brains himself. So he has built an amazing collection. And thanks to that, we've been able to study a very large number of species for now. And that's the story that you keep getting, that brains of different types of mammals and now also birds, they're all made differently.
I understand you had a great deal of difficulty in trying to get an elephant's brain through South African customs. That had to be an interesting experience. What's in the bag, ma'am? Well, it's an elephant's brain.
Actually, once we had the permits to get brains of different species out of South Africa, and that is another thing that Paul is very good with. He's learned all the ropes. He knows how to deal with that side of the bureaucracy. But I was working in Brazil at the time, and it turned out this was a really surreal conversation. I called the border patrol, the sanitary border patrol at the airport, and I told them, look, I know this is going to sound weird. I'm a scientist at the Federal University. I'm about to visit a new collaborator in South Africa, and I'm planning on bringing an elephant brain in my suitcase, and I wonder if that's all right. And this lady said, an elephant brain. I'm like, yes. In your suitcase? I'm like, yes. Is it alive? I'm like, what?
Yeah, it's a very small elephant.
No. No, it's just the brain and it's good and dead. It's fixed. It poses no biological threat. It has no commercial value. That was all that she wanted to hear. She was like, nope, we're fine. You can bring it. Not a problem. So yeah, that's all the authorities want to know. And I've since learned that it works very much the same way here in the US. If you're bringing tissue for scientific purposes, if it has no commercial value, if you have all the proper documentation, if you have the list of what you're bringing, and most important of all, if you can attest that the tissue is inert and poses no biological threat, then it's all right.
I remember from back in my undergrad days, we shipped, I'm trying to remember what it was. It's a type of coral reef fish species, and we collected their brains and sent them back. And I remember it was interesting just trying to get them to convince them that, this was okay. We weren't going to sell these. We weren't going to eat them. There's no commercial value to what we were bringing back. It was purely for scientific purposes. So
I know, right? And this
elephant
this
brain's a whole other story.
Yeah, but the funny thing is the part where they suspect that they could be edible, that's where you raise a lot of eyebrows.
Yeah.
There was one time I flew in from South Africa to Brazil, and I was bringing 2 suitcases absolutely packed with brains wrapped in, they were each in their own plastic container, sloshing around in the antifreeze that preserves them, wrapped individually in plastic bags. I had sent my husband the day before a picture of me with the 2 open suitcases where you could see all the white plastic packed packages. He was like, yeah, about that. Let me go get the bail money ready because it looks like you're bringing bales of cocaine. Yeah, but the thing is, I got to the airport and they were stopping every single person in São Paulo and putting every single suitcase through the X-ray. So I'm there calmly thinking this will be interesting, but it's fine. I'm ready for it. The funny thing was that the couple standing in line before me was coming from Portugal, and they were stopped because in the X-ray they found that they were bringing fresh cheese, and that's highly illegal. You cannot bring fresh cheese into Brazil. So they got stopped and pulled out of line. So my turn comes, and they pass. You got to remember, it's 2 suitcases, and that's all they have. It's little containers with something sloshing around in liquid. So I could see the officer's eyes just open wide, and they turned to me, "Ma'am, are you bringing fresh cheese?" And I'm like, no, it's just brains.
And they're not really fresh.
And they're absolutely not fresh. Yeah. So they had to confer and figure out who was the officer in charge. So I'm fine with that. I'm expecting it. So I just sit back with my 2 still closed suitcases. I wait for the appropriate officer to come by. And when this lady comes, I just, she starts asking me for the paperwork, and I have every single thing that she asked. She wanted a list that she could keep of every single species that I had. And I'm like, of course, here you go. Here's the list. This is your copy. These are mine, in several different languages for each different species that I was bringing. And I gotta say, I was kind of disappointed that she didn't even ask me to open my suitcases. I kind of wanted to show her the brains I was bringing.
She's thinking, I don't want to see that.
I don't want to see, yeah.
Probably, yeah.
So on that note, you've now been able to analyze the brain soup of more than 100 species. What are the key takeaways from what you found?
I think the main lesson is really that there is not a single way to put brains together. And we're working now on figuring out what the implications are. Implications in terms of how a same number of neurons, when spread more thinly over a larger surface or more thickly over a smaller surface, how does that relate to the number of modules, of functional modules that you have in a cortex? How does that relate to the cognitive abilities of a species? How does the number, you can have a similar number of neurons in the cortex, but that can actually be distributed very differently among sensory areas and motor areas and those associative areas like prefrontal cortex that we like to think are really the core ones responsible for, let's say, higher cognitive function, like pattern finding, like making plans for the future, right? So because we found that there's enormous diversity in how different brains are put together, there probably is even more diversity in how different brains work depending on how they're put together, and also how much energy different brains cost depending on how they're put together. So, yeah, if I were to put it all in one word, I would say diversity. Diversity is a huge thing, and realizing that there are multiple ways of doing the same thing, I think is really key for appreciating how brains work.
Following up on that theme, how surprised were you when you found that the elephant's brain had more neurons than the human brain? Can you talk a little bit about this, and in particular on the relative differences in the distribution of neurons?
Yeah, so the expectation when we finally were able to look at the elephant brain, the expectation was that if it's built like a regular, let's say, non-primate brain, it should still have fewer neurons than the human brain as a whole, simply because of this difference between primate brains and how they're put together with large densities of neurons compared to everybody else, right? And that's where things became really weird for a little while, because it turned out that as a whole, the elephant brain, which is about 3 times the size of a human brain, it actually does have 3 times as many neurons as a human brain. But that's the main thing. It's as a whole. And it turns out that whereas in any other mammal, primates included, humans included, that we had looked at so far, you always had 20% of neurons in the cerebral cortex, just slightly less than 80% in the cerebellum, and about 1% or so in the other structures. So 80% of all brain neurons in the cerebellum. It turned out that in the elephant, a whopping 98% of all brain neurons were actually in the cerebellum. So yes, the elephant brain has 3 times the number of neurons of the human brain, but it's really only because of the cerebellum. When you look at the cerebral cortex, it turns out that the cerebral cortex is still twice the size of the human cortex, but the elephant cerebral cortex only has 1/3 as many neurons as a human cerebral cortex. And that actually seems to apply to every single species we can look at. That includes a whale brain. The human cerebral cortex, the most distinctive feature about the human cerebral cortex is that it has apparently the largest number of neurons of any cerebral cortex in any animal, elephant included. So it turned out that the elephant was this really neat, useful experiment of nature that separated these 2 structures for us. Because unless you're willing to accept that it's really all about the cerebellum, and you would have to concede that elephants are much, much more capable than humans are, which I don't think is the case. You really have to argue for the cerebral cortex being really the part of the brain that turns all that information processing into so much more than just reacting to stimuli, than just representing sensory information and organizing responses to that sensory information. The cerebral cortex, and there's several lines of evidence that support that, it really seems that it is the cerebral cortex, aided by the cerebellum, of course, but still really the cerebral cortex that takes care of that function. So that's what we learned with the elephant, that sure, you can have a huge number of neurons in the brain as a whole, even more than in a human brain. But when it comes to the cerebral cortex, humans still rule.
So in general, would you say that the cerebral cortex is the best overall predictor of cognitive ability across species?
I think so, and the more we learn about this, the more I would say that it is, and also regardless of how large your body is, and regardless of how large your brain is, it really seems that it's all really in terms of cognitive capability. It really seems to be mostly a matter of how many units do you have to do the job, how many neurons do you have to do the job, which makes sense once you realize that a number of birds have really good cognitive capabilities on par with primates. And that makes no sense from the point of view of brain size or body size. But when you look at numbers of neurons, it turns out that they really are comparable.
Yeah, I'm curious, you mentioned the whale brain. What do we know about what we would call intelligent aquatic animals compared to land-based mammals?
Well, we know what to expect now simply because, and you have to make you have to make the difference between cetacean aquatic mammals and carnivorous aquatic mammals. But still, because they're not primates, and because the relationship between the volume of the brain or the volume of the cerebral cortex and the number of neurons seems to be really universal across the different non-primates, we can make very good predictions about how many neurons you would find in the cerebral cortex of whales, for instance. And we have studied one whale species that's not out yet, but we did find that you have exactly as many neurons as you would predict for a non-primate. And that is roughly 3 billion neurons in the cortex of a minke whale, which is, by the way, a cortex that is bigger than a human cortex. So it's very low densities.
As you previously mentioned, it was long thought that for every neuron in a brain, we have approximately 10 glial cells. And you noted that not only is this ratio high, but that cell ratio in the brain varies from structure to structure. Can you tell us what we know about this cell ratio between glial cells and neuronal cells, and what that difference means between different structures with respect to the physiology across the brain?
Sure. So it turns out that, first, in the human brain as a whole, what you have is at best a 1-to-1 ratio between glial cells and neurons. And that is important because that ratio is exactly what you would expect for the density, not the number, for the density of neurons that you have in a human brain and in the different structures. So that's the main thing that we learned about glial cells, that, first, that contrary to neurons, where neuronal densities, meaning the average size of the neurons, is highly variable, when you look at the non-neuronal cells, so mostly glial cells as a whole, what you learn is that the intuition does apply. If you take two brains of the same size, they will have similar numbers of glial cells. And if you have a bigger brain than a smaller brain, the bigger brain will always have more glial, non-neuronal cells than the smaller brain. And there you really do get this universal, almost linear relationship between the size of a brain and how many glial, non-neuronal cells it contains. What that translates into is that the density of the glial cells is essentially invariant. And to find something invariant across so many species that have diverged by tens, dozens, a couple 100 million years in evolution, that is really quite something. That was really surprising. That's evolution. The very word evolution in biology means change, not progress, just change. So to find something that apparently does not change and has not changed in at least 200 million years of evolution, that was really remarkable. So I had a lot of fun convincing people who would invite me for meetings on glial cells to let me give a talk with the title, "You Don't Mess with the Glia," because apparently that's what happens. That's what has been happening in nature. And neurons seem to be very free to vary in size and shape and morphology, but mostly really in their average size with all their arbors, but not glial cells. So anyway, so what we found was that the glia to neuron ratio seems to be really just a consequence of these two ways of varying or not varying. So where you have glial cells are added to brain structures always at the same density, so with very few changes, very little variation in their average size, whereas neurons can be very big or can be very small. So it turns out that what determines this ratio, how many glial cells you have per neuron, when you do the math, you realize that it's really dependent only on how large the average neuron is in that chunk of tissue. So the result is that where neurons in a structure that is made of few but very large neurons, each neuron comes with a lot of glial cells per neuron. But in structures that are made of a lot of tiny little neurons, then there's only a few glial cells per neuron. Now the challenge is figuring out, so what? What are the consequences? Does that mean that larger neurons can get many glial cells to help support them? How does that relate to the metabolic cost? It's really my favorite type of science, the one where you answer one question and little did you know that answer would just open up a whole new can of worms.
Right.
So you have a bunch of new questions that just come up.
Are there individual differences in neuronal density among humans? And if so, do these differences confer distinct and different abilities?
There certainly are, in the sense that there have to be. We've only studied 4 human brains so far, and I think individual differences are a really important topic, and that's something that I want to address as soon as possible. What we have done so far was look at individual differences in numbers of neurons and density in mice. And there it's really easy to collect a large number of mice that all have the same age. We've looked at all males in the case that we've published. And the interesting thing there is that you realize that the animals with the largest brains are not necessarily the animals with the most neurons. So again, you have this separation between how large a brain is and how many neurons it has. Now, having said that, I would, of course, expect that having more neurons in an individual brain should give you some biological advantage. But this is where I've realized that I had to be super careful with the words I use. That's when I started referring to biological capabilities as one thing, which I would expect to be related, at least to some level, to how many neurons you have in a structure to do that structure's job. And on the other hand, your cognitive abilities. And that is something else entirely, because that's one of the main lessons of neuroscience as a whole the last couple of decades. The abilities of a particular brain depend on so much more than simply your biological capabilities. There's all your life experiences. There's all that you train. There's a lot of self-organization that goes on in the brain. You learn to do a job the more you try to do it, the more you practice. The more positive the expectations you have that you'll be able to learn something. And this applies especially to girls in our world. The more the positive expectations that you have, the better your chances of really feeling motivated to do something and applying yourself to trying something out and finding more opportunities to just use your capabilities and develop them into abilities. So I think that's a really important thing to keep in mind when we talk about different individual human brains. Yes, I expect that there will be different biologies, different numbers of neurons, different densities, for the very same reasons that different people look different. They have different sizes. You can measure a number of differences across individuals, and not simply because they're male or female or whatever. There's biological diversity, and I think that's really important. But, and I would add, luckily, we've realized that that's really just a starting point. The opportunities you have in your life, the practice that you have, the chances you have for developing your own capabilities into actual abilities, they are so much more important, provided that you have a healthy brain.
Mm-hmm.
Makes sense.
Anthropologists such as Richard Wrangham and others have postulated a critical role for the advent of cooking in the evolution of humans. How important for our brains was learning to cook?
I think it was fundamental. I think it was really a huge divide. We've done the math, and that's when you realize that in terms of energy, in terms of the energy that it costs to run a body of a certain size and run a brain of a certain number of neurons. Remember, it's on average 6 kilocalories per billion neurons per day. If we, our species, if we still ate like other primates do, like other animals as a whole, like they do, meaning raw foods, even if including meat, we would have to spend 9.5 hours per day looking for food and eating. Forget about talking about what we're made of and how important that is. Forget about getting a college education. Forget about anything that's not spending every waking hour just looking for food. That's not viable. We know that orangutans and gorillas, they eat for a solid 8 to 8.5 hours per day, and they really can't do more than that. When food becomes scarce, they lose weight. So they can't compensate for the lack of calories by just spending more time eating per day. And that is the huge thing that cooking accomplishes. And let me just make clear, by cooking, I don't mean just using fire, which is the main focus of Richard Wrangham's points. Cooking with fire does make a huge difference, but cooking as a whole, if you think of cooking as any type, any procedure, anything you can do to modify your food before you put it in your mouth, and the really disgusting way of thinking about it, but it's absolutely accurate, is cooking is a form of pre-digesting foods before you put them into your mouth. But that's when you understand why it's really transformative, because when you chop your food, when you crush it, when you mince it, when you turn it into puree, you're actually giving yourself you're actually cutting yourself some slack. You're going to need much less effort and much less time to actively eat and ingest something that has been pre-processed already, with or without fire. So that's why cooking makes such a huge difference. You actually get many more calories from the food that you eat, because now digestion, the enzymatic digestion, will be complete. The caloric yield in your body will be much larger than what you would get from foods that you had to chew whole. You probably ingested them still in little pieces, so only the outside of those pieces can really get enzymatically digested in your body and really turn into calories for your body. So when you eat cooked foods, not only you get more calories per unit time, but you also free up time to do more interesting things with your neurons than really just looking for food and eating.
And so then that would lead to that increase in number of neuronal cells in the cerebral cortex, for example.
But because up until then, having more neurons, even if it offered some advantage, a cognitive advantage, it came with a huge cost. It was a liability. Once you get to that point where you have to devote every single waking minute that you're not just tending to absolutely mandatory businesses, social businesses. When you have to dedicate every single waking moment to eating, having a large brain actually becomes a liability. And if all of a sudden feeding that energy-hungry brain is not a problem anymore, then all you have is the benefits. Then you can really just reap the benefits of having the capability of adding more neurons to the brain with the biological benefits, let's say the increased biological capability that comes with that. So as far as I know, this is the simplest explanation for this very sudden, very steep rise in the size of the human brain that you see in our lineage from about 1.5 million years ago to now. It started with having tools that you could use to modify your food, and it, of course, must have gotten compounded with learning to control fire, to cook your foods with fire, and all of a sudden, having a large brain is all benefit.
Stephen Mithen, who wrote the review in the New York Review of Books, paid you an enormous compliment in the last paragraph of his review. He said, quote, "This is a book written with passion about a scientific quest, pursued with passion, a quest to answer a simple question that a child might ask, but that the most distinguished neuroscientists had ignored. It's given my own neurons the best workout they've had for a very long time, which is perhaps why I'm now feeling ready for a cooked dinner." Nice piece of writing.
And an enormous compliment indeed. He not only got everything right from the book as a writer who specializes in writing for the general public. I couldn't ask for any more than this, that I would be able to transmit the passion really for science and just make other people interested in the same ideas. It's been fun. So it's great to hear that other people can find that it's fun too.
And this journey that you've been on really does seem to tie back to your parents' encouragement to gather the facts and to never be afraid to question authority, doesn't it?
Yeah, I remember distinctly my father even saying to my grandma, who at some point made some comment that we should refer to her as ma'am. It wasn't appropriate to just, in Portuguese, call her by you. And I remember so clearly my dad saying, "I don't agree with you. I think that's not how you show respect. You can call somebody sir or ma'am and be extremely disrespectful. So it's not the names that you use. It's not simply agreeing or disagreeing. It's how you do it. And there's nothing wrong with asking people, well, how do you know that? Why do we think that this is true? As long as you're respectful to people and you try to understand what they're doing, I think to me it's one of the most fun things of being a scientist. It's meeting other scientists who are able to disagree with you and always remember at the same time to not take it personally. You're disagreeing with an idea. You're not saying that that person is stupid. Of course not. And that's where facts are so helpful and so important. You can talk about the facts. You can discuss the facts. I like to think that if everybody agreed with everybody, we wouldn't have scientific progress. Things would still be the same. That's something I love to tell my undergraduate students. Whenever they start getting despaired, that when I tell them that something in the textbooks is not quite right, you just see that look of despair on their faces and they go, "But what should we believe?" I'm like, great question. Exactly. So that's what you should do. Just look for the data and try to make up your mind yourself. And by the way, isn't it great that things change? Isn't it great that textbooks become wrong with time and they need rewriting? That's what science is about.
Yep, absolutely.
Well, Suzana, we greatly enjoyed our conversation. Your passion for this topic is readily apparent.
My pleasure. It was great talking to you. This is fun.
And Suzana, we'll put a link to your book in our show notes. Again, the title of Suzana's book is The Human Advantage, A New Understanding of How Our Brain Became Remarkable, which is published by MIT Press.
My favorite academic publisher.
So thank you so much again for joining us today.
My pleasure. Thank you so much for having me.
Thank you, Suzana.
STEM Talk.
STEM Talk. STEM Talk. STEM Talk.
So talk about a researcher with stories from the field to tell, a suitcase filled with brains wrapped in saran wrap, the things we do in the name of science. I loved her enthusiasm for her research. It definitely came through in our interview with Suzana.
Yes, Suzana is certainly an interesting and passionate scientist, bringing fresh ideas and perspectives into play. Also, in addition to being an excellent scientist, Suzana is a wonderful communicator of science directly to the public.
Absolutely. If you enjoyed this interview as much as we did, I invite you to visit the STEM Talk webpage where you can find the show notes for this and other episodes, stemtalk.us. This is Dawn Kernagis signing off for now.
And this is Ken Ford saying goodbye until next time we meet on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.